Hi, everyone. My name is Qin Zhu. I'm a graduate student from the University of Pennsylvania. Today, I'd like to share our study on the development of hematopoietic stem cells in the mouse embryo. So there are multiple waves of hematopoietic progenitor cell formation in the embryo. We are particularly interested in the one shown on this, on the last one shown on this graph, where the hematopoietic stem cells will form in the AGM region of the mouse. So this is an illustration of endothelium to hematopoietic transition, where the hematopoietic stem cells or HSCs will be formed. So among the arterial endothelium, some of the cells will acquire hemogenic potential and become hemogenic endothelium, and among which the hematopoietic cluster cells will rise, which contains the progenitor of hematopoietic stem cells called the pre-HSCs. So this is an confocal image of mouse embryo at E9.5. If we zoom into the dorsal aorta and Viting artery, you can see that among the many CD31 marked endothelial cells, some of them are turning green, which is marked by the RUNX1 GFP expression. These cells are the hemogenic endothelial cells, which eventually will give rise to the hematopoietic cluster cells. So at E10, if we zoom into the Viting artery of mouse embryo, you can see that the hematopoietic cluster cell form in the Viting artery and line to the vessels. So among these hematopoietic cluster cells, there will be pre-HSCs, which eventually will mature into the HSCs. So because all these populations are very rare during early development, we want to ask the question, what are the trajectory leading to the pre-HSC, and what are the underlying molecular pathways during the EHT process? So our approach is to use the single-cell RNA-seq to capture the very rare population, including the hemogenic endothelium and hematopoietic cluster cells. And we also want to purify cells based on a less purified marker, which capture the entire endothelium population, so that we know how cells are distributed along the EHT during this transition. So what we obtained is a very high quality single-cell data set, as shown in this UMAP. In this UMAP, you can see the transition from the endothelium cells to the hemogenic endothelium cells and to the hematopoietic cluster cells. Specifically, you can see that among the hematopoietic cluster cells, using known markers of pre-HSC, we collected cells at E11.5, which are the type 2 pre-HSCs. And these type 2 pre-HSCs are very far away from the fetal liver HSC at E14.5, likely because we did not capture the intermediate stages during the pre-HSC maturation. So what is further interesting about this UMAP is that we see there are two FRMB2 marked and arterial endothelium cells. They basically form two branches and they converge into this common population before they give rise to the hemogenic endothelium and later populations. So if we collect Viting artery and perform single-cell RNA-seq, we see all the endothelial cells from the Viting artery only comprise one of the branches. Whereas if we collect the dorsal aorta, there will be cells coming from both of the branches converge to the common state. And if we do differential expression, we see that the Wnt target FOXQ1 is highly expressed in one of the branches, whereas genes like HPGD is expressed in the other branch. So this pattern is very similar to the conflux of the two rivers, and we call this population where they converge to the conflux arterial endothelium. And in this single 10.5 entire endothelium batch, we can also see the convergence of the two arterial endothelium cells to the conflux endothelium and give rise to the later stages. What is interesting about this UMAP is that if we look at this place, we can see an accumulation of cells at this stage, which signifies some bottleneck blocking cells from transition into the hemogenic endothelium cells. So in order to test our hypothesis about this bottleneck, we perform RNA velocity analysis, which is based on the splicing of the RNA and the degradation of the RNA. So the velocity analysis showed us that at this step, pre-HE to HE transition, many cells at the pre-HE state have the velocity going back to the previous state. And once cells go through the bottleneck and become hemogenic endothelium, they just move forward. So this basically further suggests that there is a bottleneck during the pre-HE to HE transition. And if we look at the RUNX1 expression, we see it's mostly expressed in the hemogenic endothelium and hematopoietic cluster cells, but it's also expressed in about 7% of the pre-HE cells. So our hypothesis is that the RUNX1 is maybe a key regulator helping cells going through the bottleneck become the HE cells. So in order to test this hypothesis, we collect single cells from RUNX1 wild type and RUNX1 heterozygote and examine the distribution of cells along the trajectory. And we see dramatically decreased hemogenic endothelium and hematopoietic cluster cells and an increased number of pre-HE cells in the RUNX1 heterozygotes, suggesting RUNX1 is a key regulator governing the cell transitions through the bottleneck. And we also see that CD44 marked cells, both conflux cells, pre-HE cells, and later stages cells. So if we collect these CD44 positive endothelial cells from both RUNX1 wild type and RUNX1 heterozygotes, we see also a dramatically decreasing of hemogenic endothelium frequency in the RUNX1 heterozygotes. So what are the pathways that might regulate RUNX1 expression at the bottleneck? We performed pathway analysis and looked at the UMAP, which have the pathway activity overlaying on top of that. And we see the activation of Wnt, Notch, TGF-beta, and fluid shear stress, which are very well-known pathways that promote EHT. In order to understand the epigenetic regulation at the bottleneck, we further collected the single-cell ATAC-seq data. And this UMAP shows an over co-embedding of single-cell RNA-seq and single-cell ATAC-seq cells. So differential accessibility analysis show that there is waves of transcription factor activation during the endothelium to hematopoietic transition. And specifically, for example, in the Wnt high endothelium, we see the Wnt target TCF7 being very highly accessible in this population. And we see consecutive activation of SOC17 and GALA2 in conflux E and pre-HE. We specifically zoom into the RUNX1 because we want to know which factors might regulate RUNX1 expression. We called enhancer-promoter interactions for RUNX1, and we find many of the known enhancers, such as the +23 enhancer, is very significant based on our EP calling method. What is interesting is that we find there is another significant enhancer, 371 kilobases upstream of RUNX1. And what is interesting about this enhancer is that it is very highly active in the pre-HE stage, but loses activity thereafter. So we think this distal enhancer might be transiently required in the pre-HE to induce RUNX1 expression, but not required thereafter. So what are the cells after they go through the bottleneck and become hematopoietic cluster cells? Not all of those cells will become the hematopoietic stem cells. So what are the rest of the cells? Previously, we already showed that only the CD27 plus fraction of the cluster will give rise to the HSCs, whereas the CD27 negative part basically are the other progenitors or immature pre-HSCs. And in this study, we further show that the E10.5 CD45 hematopoietic cluster cells are lymphomyeloid progenitor cells because they give rise to B cells and T cells, but not erythrocytes and megakaryocytes. And in this UMAP, you can further see that this region, the CD27 positive pre-hematopoietic stem cells are not overlapping with the lymphomyeloid progenitors, which are CD45 positive. If we zoom into this region and do a PCA, you can see a time-dependent bifurcation of fate, where at E10.5, most of the cells will become the lymphomyeloid progenitor cells, whereas at E11.5, many of the cells will start to become the CD27 positive hematopoietic stem cell progenitors. So by differential expression, we can derive a lot of early markers marking the fate of each of the cells. Specifically, for example, MECOM is marking cells which is going towards the hematopoietic stem cell fate, whereas MEC is going towards is marking cells going towards the lymphomyeloid progenitor cell fate. So in conclusion, there are many intermediate cell types during the endothelium to hematopoietic transition, and our effort shows non-signaling pathway activities overlaying on top of the UMAP. And there is a bottleneck separating the pre-HE from the HE cells, and transition through the bottleneck is regulated by the RUNX1 expression dosage. And by joint analysis of single cell ATAC and single cell RNA-seq data, we show there is a 371 distal enhancer likely being the key enhancer for activating RUNX1 expression at the pre-HE stage. And we also show there are 2 waves of CD45 positive cells formation during the EHT process. There is an early wave of lymphomyeloid formation, and then there is a later wave of the formation of pre-HSCs. If you are interested in the details, please check out our recent blood paper. Finally, I'd like to thank people from Kaitan Lab and Nancy Speck Lab, and also Jian Kim Lab. Dr. Kaitan and Dr. Nancy Speck supervise this project. Thank you very much.
All right, thanks team for the very nice talk and very interesting new data.
Thank you.
Let's see if we already have questions. So audience, if you have questions, please type your question in the chat box. So while others may be typing the questions, Qin, I have a question for you. So you presented that you generated paired data with single-cell RNA-seq and single-cell ATAC-seq. And I think you briefly showed UMAP, which visualized some clusters. Was that the integrative analysis between these 2 types of data? And do you see any difference in the cell types that you see if you independently cluster with single-cell ATAC-seq and single-cell RNA-seq?
I see. That's a very great question. So for the integration, we actually develop our own pipeline basically to define a gene activity matrix by pairing the differential accessible peaks with the differential expressed genes. And with that activity matrix, we use the Seurat alignment algorithm to basically align the 2 data sets together. And we basically get a very consistent picture with the clustering pattern as we did for the single cell RNA-seq. And if we just use single cell ATAC-seq alone, we can also get the different cell types, which is covering the whole EHT endothelial to hematopoietic trajectory. So we basically use this kind of co-embedding to ask further questions about the epigenetic regulation of the EHT process.
So yes, I think that you developed this, is that called like gene activity matrix between the differential accessible? So so is that there? Do you kind of reduce the number of genes and the number of regions by taking only the differentially expressed genes and differentially accessible regions?
Yes. So the idea is that because traditionally people do is finding for each of the genes, variable expressed genes, for example, they look for the peaks up and downstream for a certain range and accumulate those signals as a representation activity matrix for that gene. But what we do differently is that we ask for what are the specific peaks that represent that cell type and what are the specific genes representing that cell type. And if we first do clustering in the single-cell ATAC-seq and do clustering on the single-cell RNA-seq, we can basically derive the cluster-specific peaks and the cluster-specific genes. And if the cell type from ATAC-seq corresponds to, if the cluster from the ATAC-seq corresponds to the cluster from the RNA-seq, you would expect there is the enrichment of the genomic location of the differentially accessible peaks near those differential expressed genes. And using this location mapping, we can basically correspond clusters in the ATAC-seq side with the clusters in the RNA-seq side. And then we basically can further use this link between the corresponding clusters to say this differential expressed genes, what are the differential accessible peaks regulating that gene. And we only use those really specific peaks to represent the activity of that gene. And this we find this DAP, differential accessible peak, versus DG, differential expressed gene activity matrix is actually performing much better than just using, for example, promoter or TSS signal alone.
Okay, thanks. Yeah, I think that's a good idea to bring down the number of peaks, number of regions that you need to consider for each gene. Okay, so if there are no more questions coming from the audience, I think we can conclude this session. Thanks again to all the speakers, and thanks everyone for attending this session.